Right, so today my talk is actually in two parts. The first part, I'm talking about gain from genomic selection for stem resistance in wheat. This was part of my PhD work here. And in the second half, I'm going to talk about utilizing predictions in the CIMMYT wheat breeding program and some ways we're thinking to do this and some kind of early work to determine how to go about doing that. First of all, wheat is the most extensively grown cereal crop, and this is according to FAO Stat 2012, which statistics showing that wheat is grown over more acreage across the world than any other cereal. And one of the most important diseases of wheat, especially in recent years, is stem rust. And this is especially in recent years because there's a new group of races called UG99, which overcomes the resistance in about 90% of the world's wheat varieties. And so stem rust had really not been a major concern for many years because we had resistance, but then this emerged, and you can see it came out of Eastern Africa, and it spread south and also north and is spreading across to Asia. So that this is part of the reason for the Durable Rust Resistance in Wheat project as a way to respond to this major threat. And stem rust resistance can be either simple or complex. And the simple form of resistance is based on major genes. It's highly effective, but it's very easily overcome by the pathogen because it's a gene-for-gene interaction, and so the pathogen just has to evolve one gene, and it can overcome. Then the quantitative form of resistance is generally more durable. It has a more complex inheritance, and it doesn't always give a full, complete resistance phenotype. There's usually a range. But this has really been of interest recently, especially with UG99 emerging, because we are seeing, especially in East Africa, the evolution of races is just become a lot faster. And also there's more people out there detecting races. So we're starting to really understand that, wow, this is a really rapidly evolving pathogen and we need more durable resistance. The quantitative resistance breeding is like breeding for many other quantitative traits. It requires just cyclical breeding process where you start with making crosses, generate selection candidates, test the candidates, select the best lines, and then start the process again. So each cycle you're gradually building greater resistance levels. Now, genomic selection is a new breeding technology that can increase the rate of genetic gain for quantitative traits like quantitative resistance, and mainly by reducing the breeding cycle duration. So the way it works is that with genomic selection, you can select the selection candidates prior to phenotyping. You still have to do the phenotyping, but you just can select sooner, and so that cycle time becomes less. And the way that works is you start off with a training set, which is a population that just has phenotypic and genotypic data. That's used to train a prediction model, and then that can be applied to the selection candidates in order to predict their values. And the way the prediction models work is basically by capturing genetic relationships. So we know that the more related certain lines are, the more they will resemble each other. So lines with high breeding values, their relatives are more likely to have high breeding values. And so by genotyping, we capture these relationships, and we can use that as a way to predict how a relative would perform with a certain line. And although genomic selection is not that new, it started to become popular in crops, I think in 2010. It's really gaining momentum. There hasn't been very many implementation studies in crops, though a lot of cross-validation studies, a lot of promising results. In 2013, there were a couple studies published, one in oats and one in maize. In oats, they looked at pedigree, genomic, and marker-assisted selection, but this also incorporated pedigree information, or not pedigree, phenotypic information on the candidate. So it wasn't accelerating the breeding cycle, but it was increasing accuracy of selection. On the other study in corn compared like a marker-assisted recurrent selection approach with genomic selection. So our objectives were to compare genomic selection, phenotypic selection as a way to accelerate breeding for stem rust resistance in wheat. Now, in reality, we would be selecting for all traits in the breeding program. But as a starting point, we just wanted to see, how does GS compare with phenotypic selection, looking at stem rust quantitative resistance as the trait of interest. We also want to determine if realized gain is consistent with theoretical expectations, and then look at the impacts on the inbreeding and the genetic variance. So to start off, we developed a base population for selection. So we chose it was 14 founders, did 2 rounds of intermating to establish a random mating population. Then did 1 generation of seed increase, and that became population cycle 0, which is the base from which all the selection begins. We also subdivided that into 2 replicates, so there's that way we could replicate both genotypic or genomic and phenotypic selection. And these were the breeding schemes that we followed. So we did 2 cycles of genomic selection and 1 cycle of phenotypic. And as you can see, the phenotypic selection scheme requires 2 years, where the genomic selection scheme requires 1 year. And you also can see here that the cycle 0 gets evaluated in the field, and it serves to update the training population, so that in the second cycle of selection we have an updated training set. Now the model training data comes from, for the markers, it comes from genotyping by sequencing, and the phenotypic data comes from the international UG99 screening nurseries in Joro, Kenya, and Debrecen, Ethiopia. For the cycle 1 training set, we used historical data. So this was 365 individuals that had been just breeding lines from the nursery, had been evaluated for multiple years or seasons. So there was in total 12 seasons, but each line was in approximately 4 environments. Then in the cycle 2, we used the same lines, but in addition to those, we used the previous cycle lines, for the cycle 0 lines, to update the model. So we evaluated those in Kenya for 2 seasons, used that to update the model. And you can see to the right the selection accuracies that we got in cycle 1 and 2. And you can see in cycle 1, because we're using historical data, accuracy is much lower. Cycle 2, because we updated, we have much higher accuracies. The phenotypic selection was from 2 seasons in Kenya, and we used pedigree BLUP. The pedigree is just traced back to cycle 0, so they're fairly shallow pedigrees. And our selection accuracy was extremely high, 0.82. This is a pretty highly heritable trait, so phenotypic selection is very, very effective. So, well, that font changed a little bit, but so to calculate the expected gain from selection, we use the general formula, which for selection on both sexes. So that's why there's a 2 there, because we have selection on both sexes. But the I is just the selection intensity, which is the function of the proportion selected. Then we have C, which is the covariance between the selection unit and an individual in the improved population. And in our case, this depends on the level of inbreeding in the parents. So as you inbreed more, C increases. And then we have the additive genetic standard deviation and the selection accuracy. To estimate the realized gain from selection, we took all of our populations after completing all the selection cycles and other populations and their replicates and evaluated them in 2014 in Joro, Kenya and Debrecen, Ethiopia. Then we also measured pseudo-black chaff, which is a correlated trait, which is actually undesirable because it looks like another disease. And so farmers see this in the field, they might spray the crop unnecessarily. We evaluated that in Kenya. It doesn't express, it's sort of depends on the environment, so it doesn't express in Ethiopia, just Kenya. So here is the response from selection for stem rust resistance, and on the left is the realized response. And going down means it's getting more resistant because on the y-axis is severity of stem rust, so you want lower values. And we basically get that genomic selection and phenotypic selection were not significantly different. And so in one of the replicates, genomic selection performed better, in the other replicate, phenotypic selection performed better. And we do see that there is an agreement with expected gain. So there was realized and expected were also not significantly different. So we do see quite a good agreement there with theory. Then correlated response from pseudo-black chaff was also extremely good, which is not good. we don't want that. I mean, we didn't take any measures to prevent this, but we did see a striking increase in the darkening of the heads, especially. This also agreed with the theoretical expectation. However, I think it was genomic selection cycle 1 there was one exception, so where it didn't agree completely. Then the inbreeding and genetic variance, we do see actually differences between genomic and phenotypic selection here. The genetic variance, which is on the right, we see that actually if we look at the final cycle, so on a per unit time basis, the reduction or the change in genetic variance is significantly different between genomic and phenotypic selections. Basically, genomic selection is exhausting the genetic variance faster. And this is largely because we're doing twice as many cycles. So we do 2 cycles rather than 1. And then if you look at inbreeding, we see a similar thing. This was actually not significant, so, but we do see a greater inbreeding values with genomic selection. And I mentioned it's largely due to the reduction in the time, so we're doing more cycles, but it can also be for other factors because we know that genomic selection affects allele frequencies differently than phenotypic selection. So there is actually other reasons why, even on a per cycle basis, genomic selection can lead to less genetic variance. And this is problematic because the gain from selection depends on genetic variance. So we want to keep this to be pretty high. So in summary, the realized selection responses agreed with the expected. Genomic and phenotypic selection led to equal gain per unit time with equal selection intensities. So in this case, in order for genomic selection to have outperformed phenotypic selection, we would have had to increase the selection accuracy, so our prediction accuracies would have had to have been higher, or we would have had to increase the selection intensity. Either of those two things would have brought up the gain and potentially, to outperform phenotypic selection. We also found GS led to significantly less genetic variance, and so this is really something that should be kept in mind. There are measures that you can take to try to maintain more genetic variance when you're doing your breeding schemes. So now I'm going to move on to the second part of the talk, which is the potential gain from utilizing predictions in the CIMMYT wheat breeding program. So just as some background, CIMMYT, especially the wheat breeding program, has played a key role in the Green Revolution, especially in India and Pakistan. Basically, varieties developed at CIMMYT, the Mexico program on wheat, were adopted in India and Pakistan during a time when there was actually shortages of wheat, and the yields went up, and there was basically, crises averted. And CIMMYT continues to play a key role in wheat improvement. This is just a map showing the partners, CIMMYT partners in the wheat, of wheat program that have received germplasm between 2001 and 2004. So a lot of people rely on the germplasm either as just materials to integrate into the breeding program, and in some cases the lines eventually become varieties themselves. So the current breeding scheme that's being used in the wheat program is in the first phase, it's just a basic bulk breeding scheme where you maintain populations in bulk, except there is some selection within the bulk to call out undesirable types. So things that are too tall, late maturing, resistant to disease, those get eliminated in the bulks. Then in the F4, single plants are selected and harvested separately. And in the next stage, there is about 30,000 of those F4-derived F5s that get planted in small plots. And in a lot of wheat breeding programs, they call this the hedgerow nursery, but they're actually planted in small plots. This serves for seed increase, for the yield trial in the next season, but it also gives some opportunity to do some visual selection against, again, undesirable types, or select basically biomass is a key trait that you can visually assess and select based on, just basically what you can see. Then in the first year yield trial, that's the next stage, about 7,000 go into that trial. It's just replicated twice in one location. Then the second year yield trial, it's fewer lines, so you select based on the first year data. It's about 1,200 for the second year yield trial. That is in 6 environments, all on the same station. These environments it's just different planting dates and different irrigations as a way to simulate different environments. Then after that second year of data, then lines are chosen for dissemination in different nurseries. So this is when they choose which lines get sent to which partners. And the breeding system has been pretty effective over time. Of course, so this is yield of wheat in the Yaqui Valley, which is the region where the breeding station is located in Mexico. And of course, this also incorporates some agronomic practices are also contributing to this. But the genetic yield potential has been increasing, so it has been effective. But of course, we always want to make it more effective. And so one of the opportunities to improve the breeding, the genetic gain in the breeding system is to improve this selection stage where we go from 30,000 to 7,000. Because at the moment, the selection accuracy, because it's only visual and it's on small plots, selection accuracy is probably quite low at that stage. So it's kind of this big opportunity here to improve. So my objective was to estimate the genetic gain advantage from improving selection accuracy in stage 1. Basically, if we do improve this accuracy, what kind of implications does this have Is it, basically, is it worth the effort? Is it worth the time? Then I just used deterministic simulation, which is based on the gain from selection formulas. And so basically by simulating different scenarios and then estimating gain based on these theoretical formulas, you can come up with some estimates of how accuracy at this stage will affect overall gain. So in order to do this, you have to take into account the whole breeding system, the fact that we have multiple stages and multiple pathways of selection. And so at every stage, the 30,000, the 7,000, and the 1,200, at every stage, you could potentially be selecting parents for crossing again. So that's what I mean by a pathway. So for instance, this is one pathway where we have just one stage of selection. So out of the 30,000, if we were to select parents out of that set, that would be one pathway. Then if we select parents out of the 7,000, or stage 2, then we have 2 stages of selection at that pathway. And stage 3, we have 3 stages of selection if we were to be selecting parents out of the stage 3 lines. And so because we have these multiple pathways, we have to take all those things into account. And so for each pathway, we estimate the genetic gain from that pathway. And because selection occurs at multiple stages, even within a pathway, we have to estimate genetic gain from each stage. And then the total gain for the path is the sum of those two. If there's 2 stages, if there's 3 stages. So going back to this one, since this one has 3 stages, you would have, you would look at gain from going, from the first one, gain from the second one, gain from the third one, and sum those up, and that's the total gain for that path. Also need to take into account that the genetic variance is reduced every time we do selection. So moving again from here, every time you see a red star, we're reducing genetic variance. So you have to, oops, what did I do? There. So, and there's, theory tells us what we expect the genetic variance, how it will be affected by selection. So essentially, formulas are here. Essentially, as you increase the heritability and as you increase the selection intensity, you reduce the genetic variance more. And so it's just this formula, 1 minus heritability times k, and then multiply by the starting genetic standard. This is genetic standard deviation, not variance, sorry. And k is just a function of selection intensity, where you have I is the difference between the mean and the mean of the selected in standard deviation units. X here is the difference between the mean and the truncation point in standard deviation units. So to get the overall gain from the whole breeding scheme of all the paths and selection and everything, you take the weighted average of the gain from each path. So for instance, if you took 20% of your parents coming from path 1, 30% from path 3, and 50% from, or 30% from path 2, 50% from path 3, you would use that to get the weighted average, 0.1, 0.2, and 0.5, whatever I said. And then the same goes for the time. So to get the rate, it's genetic gain over time. So you would take the weighted average of the time. So path 1 takes 1 year. Path 2 takes, or path 1 I think in this case takes 3 years. Path 2 takes 4. Path 3 takes 5. So you take a weighted average of the times, the same how you did for the weighted average of the gain. So after setting up all the different stages and different basically equations to determine the gain from these different paths, basically, I varied the selection accuracy at stage 1, and then simulated all possible variations of population size per stage, and also proportion of parents selected out of each path for each path. So you can just come up with all possibilities, simulate everything at these different levels of accuracy. Then in each case, you get the genetic gain, the maximum, and then you also take, when you find the maximum, keep track of what were the conditions, like what were the population sizes and what were the proportion of new parents at each stage. And keep track of what were those optimum values. And the main assumptions were that we had some limits on population sizes. So based on just the capacity in the breeding station, stage 1, we could probably maximum handle 50,000. First year yield trials, stage 2, maximum 10,000, and then second year yield trials, maximum of 2,000. Then some basic assumptions on selection accuracy. So from stage 1, we tested from 0 to 0.6, so looking at a range. And then stage 2, we're just assuming accuracy is 0.6, because we're actually measuring yield, but it's only based on one environment. And then stage 3, accuracy increases because now we have 2 years and we have multiple, many more environments. Then number of years, stage 1 is 3 years, stage 2 is 4 years, and stage 3 is 5 years. So we assume, of course, the trait is yield. Maybe I didn't mention that before, but yes, we assume the trait is yield, but the same would go for any trait, but we're just assuming here yield because that's the main, we're improving accuracy for yield. That's what we're trying to do. And parents can be from any stage, so any path is possible, and we make sure to take into account the genetic variance change from the different selections. So here, just the table of results. I'll walk through these things individually, but basically what we get is, we have accuracy at stage 1, then the gain at the maximum, and then we have optimum number of lines at different stages of testing, and then optimum proportion of parents selected from each path, or at each stage, essentially. So the first thing that we see is that, of course, stage 1 accuracy gives us, as we increase that accuracy, we do get greater genetic gain. But what's interesting is once we get above 0.3 in selection accuracy, we actually see a much faster increase in the rate of genetic gain. So it's sort of like as we get to a certain level, then we really start to see a big advantage. And based on these, so if we were to, say, get 0.5 accuracy compared to having no accuracy, that's about a 1.3 increase, or fold increase in the genetic gain. So it's definitely something worthwhile if we can do it. Then, so part of the reason why we see that increase once we get to a certain accuracy is it becomes favorable to choose parents sooner, so choose parents out of stage 1. So for instance, if you look here, we have proportion of parents selected on the y-axis, and on the x-axis is the stage 1 selection accuracy. So going from 0.0 to 0.3, we see that it's best to just take 60% from the final stage of yield testing, or 60% from the final stage of yield testing, 40% from the first year of yield testing. And actually, this is pretty much what is done by the CIMMYT breeding program. Without testing any of these ideas, they just basically are doing this kind of intuitively. Then once you get to higher levels of accuracy in that set of 30,000 lines that are, the stage 1 of selection, then it becomes more favorable to select parents out of that set, because we have now better accuracy. And then, so we're just kind of shifting the selection sooner, and we're reducing the time. Because we're reducing the time, we see this kind of increase at that level. Interestingly, we also see that as the stage 1 accuracy increases, it's more favorable to yield test fewer lines. And that's because, well, our selection is becoming more meaningful in the first stage. So then it makes more sense to increase our selection intensity. And so that's why the population sizes are decreased, especially from that selection where we're going from 7 or 10,000 to 2,000. So that at that point we can actually increase the selection intensity going into the first year yield trial. So that's actually really nice, because that's a huge cost and a huge effort to run all these yield trials. So we potentially can save cost here as well, at the same time increasing rate of genetic gain. So there are a couple ways that we could predict stage 1. One of them is high-throughput phenotyping and pedigree. So CIMMYT program has really, really good pedigree records. And in a lot of cases, the pedigree performs almost as good as the genomic relationship. And then on top of that, adding correlated traits from high-throughput phenotyping like NDVI, canopy temperature, we can start to get potentially really good prediction accuracies. And then there's also genomic prediction. So these two things are potential ways to get predictions of stage 1. So with the correlated traits from high-throughput phenotyping, we really find actually very good correlations with yield. So this is just from last year under optimal conditions, and we have basically 2 stages, vegetative and grain filling stage, canopy temperature, and NDVI. And here's the column with yield. And so anything that's like yellow or tan is good, and green is not good, that's about 0, and then dark blue, that's negative, but also high correlation. So that's pretty good. Most things are in about the 0.5 range, which is excellent for a genetic correlation. And then we see also pretty good heritabilities of these traits as well. This is from the same trial. Actually, the full yield trial is subdivided into like sub-trials. So each one of these squares is a sub-trial going down. And going across are the traits at different time points. So we actually have multiple time points for each. We have once per week these measurements taken. And so, especially for NDVI, we see good high heritabilities, in many cases more heritable than yield. Canopy temperature is one which seems to be a little less heritable. So in animal breeding, this approach of using predictor traits has been successful, has been demonstrated to be successful. And this was a paper about feed intake, which is a trait which is difficult to measure. And so there was, a smaller training set, and so lower accuracies. And they were looking at this as a way to improve prediction accuracy. And what this paper found was, as they increased the number of traits, the advantage of using a genomic relationship compared to a pedigree relationship actually started to decrease. So, and they, hypothesized that probably with the traits, we're capturing the Mendelian sampling term. So the main advantage of using the marker relationships was that we can get within family relationships. Well, these traits also are helping us capture within family relationships too. So then it actually, having the markers becomes a little bit less important. So here's just some comparison of, genomic selection versus high-throughput phenotyping plus pedigree for predicting stage 1. And the model that we would probably use is similar. So with genomic selection, we're still using a mixed model, the genomic best linear unbiased prediction. High-throughput phenotyping and pedigree, we still use a mixed model. It's just a multi-trait model, a multivariate, and using pedigree relationships rather than markers. We could use marker relationships, but probably it won't benefit us very much. And then some accuracies. So just from last season data, cross-validation with genomic prediction was 0.65, and cross-validation with multi-trait pedigree BLUP was 0.9. So we may be capturing a little bit more noise, or we're capturing more non-additive effects, but accuracies are basically very high with both. So either one would be good in that sense. But then when you look at the costs, we really start to see the big difference here. So to genotype 30,000 lines, or I think maybe 50,000, I can't remember if I did 30 or 50, it's about $1.5 million just to do the genotyping. High-throughput phenotyping, we've budgeted $10,000. So, I mean, that's kind of a big difference. So, and keep in mind, we still have to grow the plots, so the field cost, that's fixed, because we need to increase the seed for the yield trial. So it's still, unless technologies change dramatically, this is basically the cost one for one. So in the future work, we really need to work on trying to predict these stage 1 lines with these high throughput traits and pedigree. One of the main challenges is that we have very little seed, so there's small plots. And so all this work we've been looking at with genetic correlations, heritabilities, and prediction accuracies has been on large yield trial plots. So this is a little bit different, and it's also unreplicated. And then, so our idea is to test different plot sizes, different experimental designs. Also, what we'll do is take some lines that have already been genotyped, test them in these small plot sizes, different levels of like checks replicated, different allele sizes, and different, taking traits from different types of, cameras and other methods for getting the trait data, and just see how well we can do and what are the main factors that are affecting accuracy, if we can actually use this as kind of a cheap alternative to genotyping. So with that, just like to acknowledge the sources of funding, especially the Durable Rust Resistance in Wheat project, and also the USAID Feed the Future Innovation Lab for climate-resistant cereals. That has been really important for genotyping. They've been basically every year, the first year yield trials are being genotyped through this project. So that's been really helpful. Of course, people at CIMMYT, or obviously is the main breeder. It's been really helpful with especially getting old data and figuring out the schemes and how things work, all the costs. And there's just lots of people. Of course, at Cornell, Ronnie and Sarah for letting me go to Mexico, and Max, and then Mark, John Luke, for all their advising, and Kansas, Jesse Poland is really important for genotyping especially. So just take questions.